Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report.
Ben Zeskind likens Immuneering's platform technology to noise-canceling headphones. In fact, the company's use of the term disease-canceling technology speaks directly to that. The approach, he says, allows the company to build a pipeline of drug candidates that address aspects of disease that have eluded traditional drug development approaches. We spoke to Zeskind, CEO of Immuneering, about the company's evolution from its roots in bioinformatics, its movement into drug development, and how its proprietary platform technology works. Ben, thanks for joining us.
Happy to be here.
We're going to talk about Immuneering, drug development, and how you're using bioinformatics to cut the cost and time of bringing new therapies through to the market. Let's start with Immuneering. How do you describe the company?
Sure. Well, we're an advanced bioinformatics company. So we have a decade of experience using bioinformatics and computational biology throughout the pharma R&D process. So in the early days, that really started as services work, where we're mostly working on successful medicines and using gene expression and other types of bioinformatics data to really better understand their mechanisms of action. And of course, every drug starts with a sort of an intended or hypothesized mechanism of action, and often it's a single target. And really what we were often doing was to fill in some of the complexity around that. So in other words, to really help to say, okay, so that's the target, but why does the drug work better in some patients than others? What are the mechanisms around that? Or why, are there other targets that it's also interacting with that may contribute to the benefit? So it was really those kinds of questions that we were often working on through our services business, and that went well. We grew, company grew with revenues grew with 103% compound annual growth rate for the first 7 years. It went well commercially and also went well scientifically. We had quite a few publications and presentations. The interesting thing was, though, that we came to really view successful drugs in a unique way. I think a lot of the metaphors that people use in this field are sort of hunting related metaphors, right? They actually call themselves drug hunters. You talk about targets and hits, and it's there's sort of an element of chance, an element of trial and error or serendipity to kind of happening onto something. And what we came to see in our data, and particularly in looking at a number of successful drugs at the gene expression level, the RNA profiles that they induced, we came to view successful drugs more as signal inducers than bullets. And so, again, sort of the traditional thought was you sort of take out one specific target like a bullet would, and then even some of the newer methods such as gene therapy or CRISPR. It's sort of like a narrower, more precise bullet. But for us, and when we looked at it sort of bioinformatically at the gene expression level, the successful medicine, the most successful ones looked more like kind of signal inducers than kind of bullets. And based on that, we really came to view, we developed a kind of a different metaphor for successful medicines, which is actually the noise canceling headset of all things. So, I think most people are familiar with getting on a plane and you turn that on and the noise, unwanted noise kind of almost magically vanishes. And really the way that's working, and it appeals to us as engineers, half our team is trained at MIT. There's a microphone in those headsets and it's listening for the unwanted noise, and it creates an anti-noise. In other words, sort of a mirror image of the unwanted noise, a signal that's 180 degrees out of phase, and it puts that together with the noise, and you get destructive interference, and it cancels it out. And that's really kind of how we saw successful drugs to be working at the gene expression level. And as a result of that, we set out to build a technology, a computational technology that would let us create new medicines sort of with that in mind from the start. So kind of engineer drug candidates that would effectively cancel out at the gene expression level an aspect of disease that we're looking to treat. And we spent several years building that technology and refining it and validating it. And it started doing quite well, at which point we set out to develop our own pipeline of drug candidates, and we call it disease canceling technology, by the way, for hopefully obvious reasons given the metaphor. But so we once we had developed that disease canceling technology, we began developing a pipeline of wholly owned drug assets using it.
Let me take a step back. You mentioned
Sure.
the MIT pedigree of your staff. You've got a background in electrical engineering and computer science. How did you come to drug development? What was the opportunity you saw?
Yeah, it's a great question. Thanks, Danny. So you're absolutely right. My undergraduate training was in electrical engineering and computer science at MIT. That's what my father had studied, so I was kind of inspired by him. Being at MIT at the time that I was, it was right around the time of the Human Genome Project. So there was really just a lot of excitement about genomics and what it could represent. So I actually, as part of we had a lab requirement in one of my electrical engineering optics classes, and through that had to do a project in a lab at MIT. So I chose a microscopy lab at the Whitehead, doing biological microscopy, and that just really loved that. Loved sort of the intersection between engineering and biology. So I ended up staying at MIT to do my PhD in bioengineering in that very lab. Paul Matsudaira was the principal investigator, and the department, the biological engineering department at MIT was really set up for kind of people like me, people who were coming from engineering backgrounds and wanted to apply those insights to biology, the sort of quantitative approach of thinking about problems and thinking about signals and systems, and the department was really built and led by Doug Laufenberger, who's on our scientific advisory board to this day. So really through that training at MIT, I saw the power of kind of taking a quantitative engineering mindset and applying it to biology. So, when I finished grad school, I ended up doing an MBA. I knew I wanted to start a company, but not exactly on my thesis research. So it was actually a good opportunity to take a step back and figure out, where exactly I thought the right opportunity was. So, during my MBA was really fortunate to meet my co-founder, Bob Carpenter, who has started dozens of biotech companies going back to integrated genetics in the early 80s. He was on Genzyme's board for nearly 2 decades. So met him, and really the need that we identified at that time was just that the ability to generate high throughput biological data, gene expression, DNA sequencing, proteomics was really outstripping or outpacing the ability to make sense of it and turn it into useful or actionable insights, particularly for the development of new medicines. So the costs were plummeting, and we saw that all the pharma companies were generating just increasing volumes of this kind of data. At the time, it was a lot of microarrays. That was the technology for gene expression. Now, of course, it's mostly RNA-seq that's used. And there were SNP chips and just all this data, and there weren't as many groups that were really focused entirely on analyzing the data and sort of getting the most out of it. There were at the time those kind of spaghetti diagrams were popular. That I don't know if you remember. They kind of looked like a hairball or a I think there was a quote in Nature, an explosion in a spaghetti factory. But it was just these plots that would show how every single gene was connected to every other gene, and it was sort of it was nice art, but it wasn't really necessarily something a pharma company could use to make a decision. So we really said we're gonna focus entirely on the analysis of the data and extracting more insights from it that could be meaningful to drug development, and that was really kind of the motivation around starting the company.
You mentioned your computational platform that you call disease canceling technology. What exactly is the platform, and how does it work?
Sure. Yeah. So that of course was developed much later on, as I mentioned, and it's an algorithm. So it's a computational technology, and really it takes as input a gene expression signature associated with a particular aspect of disease that we're looking to cancel. So one of our drug programs is for cancer cachexia, which is the wasting of muscle tissue that occurs in late stage cancer patients. Many people are familiar with the kind of emaciated appearance that can occur, but fewer people know that it's actually the final cause of about a third of all cancer deaths, because sadly the heart or the lung muscles become too weak to function. So this is kind of the final cause of a huge number of cancer deaths, and yet there are no approved therapies for it. And I think a big part of the reason for that is it's just not amenable to traditional drug discovery methods. So kind of traditional drug discovery, a lot of times you're screening targets or drugs in vitro in cancer cell lines in a petri dish, if you will, and really looking to see how well you can kill the cancer cells. And it's measured often with an IC50, and it's that's often the focus, and that's good for discovering certain drug candidates, but for aspects of disease like cachexia, where it's the result of a complex set of interactions between the tumor and the immune system and the patient's muscle tissue. Screening in cancer cell lines in vitro is just not really going to discover an effective medicine for that. So I think that's where platforms like our disease canceling technology can really shine, because so what we're able to do is to take gene expression data from muscle tissue of cancer patients with and without cachexia, as well as relevant model systems, and really compare them, using careful careful bioinformatics, including very rigorous quality control and normalization. And what that gets us is a really precise signature specifically for which genes are changed as a patient develops cachexia. So that's kind of the key input to our platform. That's the disease signal, if you will. Go back to the metaphor. That's the unwanted noise that you're looking to cancel out. And then with that input, what our platform is able to do is really two steps. And again, it's computational. So there's a biology step and a chemistry step. And in the biology step, what we're doing is essentially algorithmically ranking targets by the extent to which they are able to cancel out that disease signal. And we do that in a unique and proprietary way. And then the second step is once we have the target, our platform is able to essentially do a virtual high throughput screen, so a chemistry step where using the amino acid sequence of the target, we're able to screen large libraries of tens of millions of compounds and identify the predicted binding affinity. So identify predicted hits. And then to be clear, of course, we have to experimentally validate that downstream. But so we're with the platform, we're able to computationally identify sort of both targets and hits, but ones that are not sort of solely for that target itself, but are targets that we know are relevant because they induce a whole set of signals that are likely to cancel out the aspect of disease that we're looking for. So that's really the disease canceling technology. It's really a biology platform based on gene expression, closely coupled with a chemistry platform that gets us to the hits, the initial chemical structures that we then, of course, have to experimentally validate and lead optimize, in a more traditional way. So that's really the technology.
Why do so many drugs fail in discovery and development, and how does your approach minimize the time and money invested in advancing molecules that won't pan out?
Yeah, it's a great question, and it's a real challenge. I think the productivity in pharmaceutical R&D is certainly has a lot of opportunities for improvement. And there's a number of reasons that drugs fail. So we can talk through some of them. I mean, I think the first is that traditional drug discovery methods often require starting with model systems. So you're doing the initial work in either cell lines in vitro or animal models, and those are important systems, and they're useful for discovering a lot of drugs. But as I noted in the cachexia example, there's a lot of aspects of disease that they either can't model or don't model accurately. And so one of the nice things about our platform is that with the disease canceling technology, we can actually start with human patient data. So the very origin of our drug programs, when we're doing that initial biology step, is based on human patient data. And so I think that's an important factor for the ultimate success of our programs, because while of course we have to do the same, do experiments in model systems as well, I think there's a difference between starting with a model system versus starting with human data. So I mean that's one of the ways that we're able to increase the rate of success. And then I think the approach of disease canceling technology in terms of looking across multiple, looking at the impact of both a disease and then the drug candidate across tens of thousands of genes. Oftentimes drug candidates will fail due to sort of unintended consequences. off-target effects or polypharmacology. And in our case, we're kind of leveraging that from the start. We're taking that into account from the beginning and sort of engineering it in. So I think we're able to do a lot of that. So that's in terms of the success rate. In terms of the time and capital efficiency, it's a little more straightforward. So most frequently the two steps, target identification is frequently done using trial and error experiments that often rely on serendipity, right? You have to kind of stumble upon something or kind of get have a lucky break. And then similarly, high throughput screening, you're running a lot of experiments. You're essentially testing many molecules in the library experimentally. So both of those steps, both have a serendipity component and just require months and months and significant dollars to run the experiments. And we're able to do most of that computationally. So just the switch from having to do that experimentally to computationally is a pretty significant speed up and saves a lot of capital. And we kind of anticipated this, but it wasn't really until we had our drug programs up and running and we had kind of six programs in our pipeline that we actually went back and looked at our historical actual numbers. So that, not projecting, but just based on our data from our programs to date, and compare that to benchmarks. And we're about twice as fast and about 3 times as capital efficient as sort of a traditional benchmark.
Is that referring to the discovery phase, just getting to clinic, or is that for the entire development?
That's from inception to proof of concept in animal models. So it's really on the front end of the process.
And is that, I know you've been a service business for about 10 years or so. Is that what the data is based on, or is this actually in regards to your own drug discovery?
Right. Yeah. So this is in regards to our own internal pipeline, and I should just clarify. Our services business continues to this day, and it's thriving. It's a little bit different though. So we actually don't use the disease canceling technology or other proprietary platforms in our services. So our services business is really just kind of advanced bioinformatics, sort of best practices. Computational biology for analysis of existing data sets that different pharma companies have. So it's pretty separate in that sense. I think the thing that unifies it all is just this foundation of being kind of second to none at bioinformatics and in the kind of rigorous reproducible analysis of bioinformatic data.
And is that business able to fund your own discovery and development?
So it certainly has, and in fact, I think one of the things that surprises people is that we're, our company is 11 years old, and we just raised a Series A.
It's a long path to a Series A.
Yeah. So, but actually, it's great because it means that we were able to fund all the early work, seed all the early work off the balance sheet from the services business. And so that's, it's actually been really, really helpful. So yeah, and but again, there's, it's really just the unifying factor is the common experience in bioinformatics.
You mentioned the cancer cachexia program. What's the target in that indication? So based on our disease canceling technology, we actually identified a couple of related targets that our drug is going after. But one of them that we've disclosed is MEK. So MEK is one of the targets of our drug, and we actually presented data on that just a few weeks ago in Berlin at it's called SCWD, the Society for
cachexia, sarcopenia, and muscle wasting.
So is this a tyrosine kinase inhibitor?
I don't want to get into too much detail on how the molecule's working. We've not disclosed that, other than to say that MEK is a target, which of course is a kinase. So it's not an unreasonable question for you to ask.
Well, you've got, I think, about a half a dozen other programs in preclinical development right now.
That's right.
This gets expensive once you start moving to the clinic. What's the plan on prioritizing the programs and the indications you're pursuing?
Yeah, that's a great question. So, in terms of prioritization, we're certainly very data driven. I think our focus is on disease areas where our platform can identify medicines that sort of weren't previously possible with traditional drug discovery methods. So, we're really focused on areas specifically where where our platform can create medicines where there really aren't any, or sort of aren't the right ones. You're certainly right that as you progress along in the drug development process, the expenses go up, and we're certainly excited to take our some of the drugs in our portfolio through to clinical trials. And in fact, we recently hired a new chief medical officer, and we're excited about that, and he'll be helping to drive these programs through to the clinic. But the interesting thing is, because of the capital efficiency with which we can bring programs to the point of in vivo proof of concept, as I mentioned, we're about kind of 3 times more capital efficient than benchmarks. But there's another aspect to it, which is that we have a much steadier stream of new assets. So and this has to do with eliminating some of the trial and error or the serendipity. So I think a lot of biotech startups have to focus on a single target or a single pathway, because just kind of the years of experiments have sort of pointed them in that direction. Whereas for us, with the disease canceling technology, as long as we have a good gene expression signature for the aspect of disease we're looking to cancel, we will get targets and hits. That doesn't mean they're all going to work, and we certainly assume a healthy failure rate. But just the steadiness of that output, I think, is very different from kind of traditional biotech startups. So it's the combination of those two things: the steadiness of the output, the steady stream of drug candidates we're able to create, and the capital efficiency with which we can do it. It really creates, in some ways, kind of a new business model, because the types of upfront payments that pharma is typically willing to pay at after in vivo proof of concept, while they wouldn't be an attractive exit for a kind of a company that has a single shot on goal or a small number of programs. For us, with the capital efficiency, it's actually an attractive ROI even at that point. And because we have a steady stream of other assets, it's not as if we're sort of giving up the limited number of assets. So we actually are pursuing a kind of a hybrid business model where some of the and we kind of make the decision on a program by program basis. So we're certainly equipped to take any program that we want all the way through to the clinic. But on a program by program basis, where we find the right partners, we're also open to licensing drug candidates earlier on, sort of shortly after in vivo proof of concept, and we can do that at an attractive return on investment on a per program basis. So that's kind of I think a fairly unique business model that's enabled by by our disease canceling technology.
Ben Zeskind, co-founder and CEO of Immuneering. Ben, thanks so much for your time today.
Thanks for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.